everybody. I see only 9 students here today, but I will get the class started anyway. So if you get a chance to talk to other students, such as in preparation of your presentation, please let others know that I do, emphasize class attendance, lecture attendance. So if you are not going to be here for, significant amount of your lectures, don't expect to get an A from me. I expect attendance, good attendance, not necessarily every class. Occasional missing of a class is okay, but not frequent missing. So I'm gonna continue the discussion of somatic cell nuclear transfer today. But on the first slide, as you can see, there is a scientist there. Can somebody, try to guess who he is?
Darwin.
Great, he got it on the first try. So this is Darwin. People consider he's one of the, probably not one, he's the most famous biologist in the history. But he, during his time, he suffered some strange disease. He has frequent muscle spasm, and also vertigo, dizziness, vomiting, gut pain, things like that, a lot of problems. So at that time, there were about 20 doctors who ever treated him. They feel like his problem may not have a physical origin. Maybe it was from his mind or something like that. And later, when he was dead, after he was dead, and people still trying to figure out what could be the problem. A few years ago, there was a graduate student published a paper said maybe Darwin had lactose intolerance. Lactose intolerance, you must have learned that in biochemistry, is because people doesn't have the enzyme that will break lactose to easier monosugar, so that the body can absorb and take that as a nutrition source. But later on, other scientists found that maybe Darwin suffered from a disease called mitochondria disease, because similar symptoms was present in his mother as well as an uncle from his mother's side. And I couldn't remember exactly the name of the disease, but they even pinpointed the mutation, the possible mutation there for his disease. So that mutation is called A3243G. With that name, you can predict that is alanine that changed to glycine, and the 3243 is, it's just a identifier of the mitochondria gene, one of the mitochondria genes. So today I'm going to describe a new approach in treating mitochondrial diseases, which uses the principle we learned in the previous lecture, somatic cell nuclear transfer. In this case, it's not necessarily somatic cell nuclear transfer, it's just the nuclear transfer, but the principle stays exactly the same. I think this is very interesting. That's why I kind of insert this part into our lecture series. It is not in our textbook. So first of all, I would like to briefly tell you a little bit about mitochondrial disease. It is a very common disease. It happens in 1 out of 4,000 children. That's that common it is. And about 15% of mitochondrial disease are caused by mutations. The mutations are on mitochondrial genes. So I don't know if you learned that elsewhere, but the nucleus is not the only place you find genetic material. So of course, the majority of our genes are encoded by the nucleus, by the DNA there. We call it the genome. I would say maybe, I don't have an exact number, but must be more than 99.99% of genes are located in the nucleus. But there are genes in the mitochondria as well. So in the mitochondria, you also have genes that encode RNAs and proteins. You hear I frequently say that genes encode for RNA and proteins because nowadays we know the genes not only encode for proteins, but also some RNAs that will not get translated to protein. So those RNAs are also functional. So that's why I say genes for RNAs and proteins, not only genes for proteins. So in the mitochondria, you have genes that encode for small amount of RNAs and protein species. And those proteins and RNAs actually perform very important function in the mitochondria. So if there are mutations in the mitochondria genes, a lot of times there will be some health issues. And often those health issues are quite severe. And as you know, the mitochondria is a metabolic organ, right? It's not organ, I'm sorry, it's a organelle. And then usually those diseases are related to metabolism. In the mitochondria is so-called the energy factory. So if somebody have problem in the mitochondria, usually it's related to something like a diabetes, for example. So here I give you examples of mitochondrial disease. One of it is called diabetes mellitus and deafness. It's not the diabetes, the common diabetes we frequently encounter in our relatives because it is a quite common disease. And here this DAD is a kind of a genetic problem. Is because the mutations in the mitochondria. And mitochondrial disease are inherited from the mother. Why? You will see soon that during fertilization, the sperm carries in, roughly speaking, just the genetic material. The egg is so big compared to the cytoplasm of the sperm. So the majority of the cytoplasm in fertilization is from the egg, is from the mother side. Mitochondria is in the cytoplasm, as you know. Therefore, the mother just pass on majority of the mitochondria to the kids. So that's why mitochondrial disease is inherited from the mother. Okay, so that's why I say at the beginning when people observed that Darwin's mother side uncle and his mother also suffered similar symptoms, so they feel like maybe he had mitochondrial problem. And although this is really common and severe, there's no treatment. So you can only have some vitamins or other things that can support relatively good quality of life, like painkillers, things like that. There's no actual cure of it. Nothing can, to remove the source of that disease. Nothing can correct the mutation. Maybe in the future we can, but at this moment still there's no treatment. But UK, I believe it's already available. They have, developed this approach to treat mitochondrial disease, which is the topic of today. I'm going to give you, the preclinical test in animals, how they try to treat this disease using nuclear transfer. And then just one more words about this, why UK is so advanced in this type of research is somehow UK is more open in stem cell research in embryo manipulation than US. Here, you can imagine this kind of approach will be even explored. Even in preclinical animals, it's not explored. But somehow, UK is more advanced, and they have a lot of policies that actually encourage stem cell research, human stem cell research. So, but first of all, let's go back to the early stage of development again, fertilization and meiosis. So for you to understand the technical part of that IVF manipulation, let's review these things again. So the homologous chromosome recombination occurs at prophase arrest. This prophase 1 arrest. Okay, the prophase 1 is okay. This is when the oocyte is still in the ovary, as they are arrested in prophase, and then you have spindle assembly. That means the cells are ready to get divided, and then once you complete the first meiosis, it's going to generate a polar body. It's the first polar body, and the spindle itself. So the cells get arrested again, but at this time, usually this time is right around when the oocyte is released. That process is called ovulation, right? Called ovulation. Basically, around ovulation, right before and after that, the cell division will be completed, and then the cells are arrested at metaphase 2. So you have the chromosome already condensed. That's metaphase. The chromosome is condensed. We call it a karyoplast. So that's or you call it a spindle. So that's the nucleus of the oocyte, but also you generate a polar body. I call this polar body 1 because later on, once this metaphase 2 arrested oocyte is fertilized, and then the second meiosis will become completed. At that time, another polar body will be generated because the oocyte will complete another meiosis and generating a smaller cell that's called the polar body. So you have two polar bodies, two types of polar bodies. One's because of the first meiosis, the other is because of the second meiosis. And the first polar body is at the stage of ovulation. So when the oocyte is released, but still before fertilization. So that's when you will see the first polar body. And the second polar body is after fertilization. It involves a male. Okay. Make this very straight. Otherwise, you won't be able to understand what I'm going to say later in this lecture. So this is what they're trying to do. So I'm going to describe the contents of a paper, and this is the outline of the anti-experiment they are trying to accomplish. So basically, the goal here is, okay, there's this mitochondria disease. So this mom have mitochondria that have mutations, right? So you try not to use the mitochondria of the mom, but you still want this mom to donate the genetic material, to the offspring, to the kids. So the goal here is to, try to avoid the mitochondria from the mom with mitochondria problem. So therefore, what you are trying to do here is take the genetic material from the mom oocyte. Okay, from this problem mom oocyte. You take some genetic material from the oocyte, and now you put into another healthy egg. Okay, in that healthy egg, you take out the nucleus first. So just like in somatic cell nuclear transfer, you have a donor egg. You have, I'm sorry, I don't want to use donor because in this case, it's very complicated. Which one's donor? You will see why it is hard. So I would say, okay, this healthy oocyte is from a source. Somebody donated this healthy oocyte, but you remove the nucleus because you want the babies to still be from the mom that have that mitochondrial problem. You wanted the genetic material from that unhealthy mom, but you put that nucleus into a healthy oocyte. That's what they're trying to do here. Okay, but they're testing 4 different possibilities. But first of all, in every occasion, you start with enucleation of recipient oocyte. See why I don't want to use donor to describe the healthy oocyte, because that's actually the recipient oocyte. Okay, so first you remove the nucleus from the recipient oocyte, and then we're trying to do 4 different manipulations. Okay, in one manipulation, you take the polar body one. Okay, the polar body PB1 from the problem, the oocyte with problem. Okay, now this is the donor oocyte. That's, you have this red mitochondria which have mutations. So you want to avoid the mitochondria. So you take the polar body. The polar body will have genetic material just like the oocyte itself. It's the same genetic material, right? The DNA sequences, the copy numbers of DNA are going to be the same. So but you take the polar body, you put into the enucleated recipient oocyte. That's what you do. That's the first occasion, the first thing they want to try, first condition they want to try. And in the next condition, what they're trying to do is, now they don't take the polar body, but instead they take the spindle itself. The spindle, of course, is the monocyte nucleus. It's the monocyte nucleus, right? Although at this stage there's no nucleus, right? Because nuclear membrane is broken. It's spindle, but the spindle is the genetic material. So you just take the spindle. Now you put the spindle into that enucleated recipient oocyte. As you can see here, you're trying to use the genetic material from the unhealthy mom, but you're trying to use the cytoplasm of a healthy woman. Okay, that's what you're trying to do. I explained to you 2 conditions they want to try, and then in the third condition, what they're trying to try is they allow this oocyte, it's from the unhealthy mom, this oocyte to get fertilized. So the dad is involved as well. So the dad will, give the sperm will fertilize this oocyte, and then they allow this to develop. Of course, that's almost instantly you're going to have PB2, that's the polar body 2. Once fertilized, the oocytes will generate another polar body. Now, you can take the second polar body, and now you put into the enucleated recipient zygote. Now you see the name changed. It's zygote because the oocyte is fertilized, and now we call it zygote. Okay, the recipient, of course, will be the zygote as well, because the developmental stage should match exactly to each other, right? Or instead of taking PB2, polar body 2, you can take the pronucleus, the pronuclei. At this stage, there are 2 pronuclei. one from dad, one from mom. Two nuclei here. So for that transfer, you have to take both nuclei, like shown here, and then you put into the nucleated recipient oocyte, healthy oocyte. And in all the occasions, you just allow that oocyte, the manipulated oocyte, to go through blastocyst and then go to give a birth. And then later on, because this is animal experiment, you can do further mating to make the so-called F1 pup. You know, that's the children of this manipulation. You can allow that those pups to grow up and eventually give rise to this another generation. So use those grown up pups to mate and then give rise to F2. Okay, that's the next generation. And you study whether F1 and F2 have a mitochondrial problem. Okay, that's the principle here. Do you have questions? I want to pause here. I can listen to your question and answer you. Okay, I hear no questions, so I'm going to move on. But on the first slide, of that experiment, they described where the mitochondria is. So in the cells, where you find the mitochondria, because you don't want mitochondria carry over. You're trying to take the genetic material and then you put that genetic material into a healthy oocyte or zygote. So you don't want any mitochondria to get carried over, right? You don't want to take the mitochondria, you know, as a byproduct into the oocyte. So you want to study where the mitochondria are distributed. So here in A panel, what's shown is the oocyte stage. You only have PB1 because the oocyte is not fertilized yet. So you have a M2, that's metaphase 2 chromosome, you know, that's the spindle shown there. And then you have the PB1 outside of that circle. The circle is the egg. So the PB1 is the polar body 1 is outside of that cell because you have two cells. One's a big egg, the other is a much smaller polar body. So you see here is a Hooke's staining. The Hooke's staining is, you learned already, this stains nucleic acid. So those blue colored spots where the DNA is. Okay, so you see the nucleus of the oocyte, and you also have the DNA of polar body 1. And in the next staining, what you have is staining of metal tracker. By name, you can predict this is a chemical that's preferentially bound to content in the mitochondria. Now, it reviewed that the oocyte have a lot of mitochondria, almost everywhere. You know, in the oocyte cytoplasma, you see mitochondria, but it's almost excluded in polar body 1. Polar body 1 is just a smaller cell. You don't see much mitochondria there by the staining. Okay, so that's what happened in the oocyte. And the bottom here is just another example, just to show you that you have a lot of mitochondria in the oocyte, but much less or almost none in the PB1. So that's what happened in oocyte. Okay, in B panel, what you see is what happened in zygote. So now this embryo is a fertilized egg. After fertilization, you have two nuclei and a polar body 2. Okay, and now if you stain with Hooke's, what you see is 2 pronuclei and PB2, polar body 2. And if you stain with metal tracker, again, a lot of mitochondria in the zygote at this stage, just like in oocyte. But if you look carefully, you see that in these two examples, you know, if your room is bright, you can turn off your light to see clearly the fluorescence image. So here you can see in polar body 2, it still have a lot of mitochondria in there. So in polar body 1, you have much less mitochondria, but in polar body 2, in these two occasions, you see some mitochondria in there. Okay, so you see that's when they take the genetic material out. So in A and B, that's when everything is still intact. You didn't do any manipulation. In C, it's when you take those, you isolate the polar body 1, polar body 2, spindle, and pronuclei. You take them out. You put into a pipetter, or put into a culture vessel in which you can take a picture. And now let's see the carryover. Okay, that's carryover. When you take them out, do you have a mitochondria in there? In PB1, well, with mitotracker staining, maybe there is a little, some red spot there, but it's the least among all the manipulations. The second manipulation, that's when you take the spindle. You take the spindle, you try to take the spindle here, but the only way you can do it is to put a micropipetter in there, you try to get it out. So carryover of mitochondria is hard to avoid, right? Because you take that mechanically. As you can see, in the spindle itself, it carries over a lot of mitochondria. And when you take out PB2, you try to just take the PB2 nucleus, but you also have a lot of carryover of mitochondria. And when you take pronuclei, that's this thing. You try to mechanically take the pronuclei, you carry over a lot of mitochondria. So based on this information, you can kind of predict maybe, using PB1 have the greatest advantage. So you can predict that, but let's see if it is true. But first of all, this slide is kind of unrelated to what I'm going to describe later, but I want to, because students said you're interested in this thing, so I want to tell you DNA methylation during early development. So the paternal, of course, is the sperm, and maternal is the oocyte, right? So before fertilization, if you check the DNA methylation level, you can see that the paternal side have higher level of DNA methylation. So you have this indicator called 5-MC. It is 5-methylcytosine. Okay, so cytosine 5 get methylated. That's why what you call it 5-methylcytosine. DNA methylation. Okay, this is not about histone, but only about DNA methylation. The DNA methylation suggests that if you have a lot of DNA methylation, that means maybe that region is not open. Okay, relatively speaking. So when you have less methylation, DNA methylation, that means that region may be more open. The gene expression will be more active. So you see in the paternal side, the methylation at the very beginning is high. Well, from the maternal side, the methylation is low. But after fertilization, the methylation on the paternal side, that's the sperm DNA, right? It dropped very, very rapidly. So quickly, the methylation is erased. You don't have much methylation. But in the maternal side, it also drops, but much slower after fertilization. Okay, so after fertilization, you can predict that the paternal DNA methylation is minimum, while you still have some DNA methylation on the maternal side. That's what happened in oocyte, right? I'm sorry, in zygote. I should call zygote after fertilization. But they will stay low during early development, like when the cell become 2-cell, 4-cell, 8-cell, and morula, DNA methylation is only minimum, as you can see there. But when the cell becomes blastocyst, DNA methylation start to pick up in both maternal and paternal DNAs, because you have two sets of chromosomes, one from mother, one from dad, but the methylation will occur, to similar degree in both maternal and paternal in that blastocyst stage. So why I said that is because I want you to appreciate the scientists are trying to use that information to distinguish what is what is the maternal DNA, what is the paternal DNA. So on this slide, I want you to skip D and E panel and just to focus on the rest. So let's first study F and G. So basically, this is what the manipulation will be like. So remember, this is oocyte. The developmental stage is oocyte. So you have this PB1 there, in that black and white picture. You have a bigger pipetter serving as a holder. It sucks it have a negative pressure there. It sucks the oocyte to fix the oocyte in place, and then you use another much finer pipetter, push in, and you get the PB1 isolated, the polar bottom one isolated. And next is just to push that. You push that pipetter, the same pipetter with the polar bottom one in there. You push into the recipient healthy cytoplasm of an oocyte. You push that polar bottom one in there. This is a nuclear transfer. Although we don't, this is actually not a somatic cell nuclear transfer, because at this stage it's germ cells. It's not somatic cell, stem cell actually. You push that. That's what you do for PB1 transfer, and then in G, this tells you what they did for a spindle transfer. Okay, so instead of taking the polar body, you push the macropipetter into the cytoplasma, and now you try to take the spindle itself. Okay, that's what you're trying to do. You take the spindle itself, and now you push. You take out that pipetter, and now you put that spindle into a healthy recipient oocyte. Okay, that's the two manipulations you are trying to do. So now let's focus on A, B, and C, see what they are trying to observe. So in A and B, they stand for alpha tubulin. Alpha tubulin is a component of spindle. So when you have the centrosome, right, the centrosome will pull the spindle to opposite side of a cell. So tubulin is part of the structure. So when the spindle will tell you, the chromosomes in the process of getting separated. So you can see that this is PB1. You have spindle in PB1. You have the spindle in the nucleus as well. So when you stain the chromosome with a red dye, you can visualize that the chromosome is located at the equator, right? It's because the cell is on metaphase arrest. So the red color indicating that the chromosomes are located right there at the equator. So this not only tells you where the polar body is, where the spindle is, but it also tells you, okay, this is arrested embryo here. The oocyte arrested at metaphase. Okay, that's why they do this two staining of a tubulin and a chromosome staining. And then next, I want you to pay attention to 5-methyl cytosine. That is a methylation of DNA, cytosine methylation. Remember, let's go back to the previous slide. This is before fertilization. So the maternal side still have quite a lot of DNA methylation. That's why you see the DNA methylation marker present in both polar body 1 and the spindle itself. Okay, so because the methylation is still present there in the maternal before fertilization. So this is the first manipulation they're trying to do, the first two manipulations they're trying to do. So they're trying to transfer polar body 1 or the spindle into a healthy recipient. So in this slide, I'll describe to you the third and the fourth manipulation they are trying to do. Of course, these four manipulations are in parallel, not sequential, but in parallel. Again, you don't need to focus on C and D, just A, B, E, and F. So in E, remember now it's zygote, it's fertilized egg. Both the unhealthy oocyte and the healthy oocyte are in the same developmental stage, that's zygote, fertilized egg. In fertilized egg, you will have two nuclei called pronuclei, but also you have PB2. As shown there, you have a PB2, and you can take the PB2 from the unhealthy donor, and you push that PB2 into the healthy recipient. Okay, into the healthy recipient. Next, the fourth manipulation. In the fourth manipulation, of course, you're trying to take the pronuclei and now put into the healthy recipient. Okay, but remember here, I think that was there's a subtle difference there. So the PB2 may not be the desired manipulation, because in the recipient, you also, your recipient, you also have the recipient genetic material in there. Okay, that's just some nuance. But aside from that, what you're trying to do is transfer the genetic material from the unhealthy oocyte or zygote, and now get that into a healthy recipient oocyte or zygote. And now let's study two stainings here. So one is lamine staining. Lamine is component of nucleus. So this will visualize where the nucleus or nuclei are. As you can see, it stains polar body 2 and 2 pronuclei. So even the nuclei size are very different. The mom side is quite big. Well, the paternal side is I'm sorry, I got it wrong. The mom side is the same size as the polar body, because the after cell division, you generate a polar body 2 and the mom side oocyte. A mom side still oocyte, yes. So the size of the nuclei are the same, but the paternal side, you get a relatively bigger nuclei there. The reason is because one's more compact than the other is looser. Okay, because the genetic material there, the chromosome numbers, DNA amount are the same. It's just one's more compact, the other is looser. Okay, but that's why when you stain for chromatin, you see darker staining of the mother side nuclei for polar body and for the mother side, oocyte. But because the structure is looser, chromatin staining will not give you an intense image of the bad side, nucleus there. And now let's observe 5-MC. 5-MC indicate DNA methylation. This is a marker of DNA methylation. So let's go back to that illustration here. So the paternal side after fertilization drops very, very quickly in terms of DNA methylation. So now let's look if that is the case. Now let's see. You have the chromatin staining that shows you the 3 nuclei here. One's the polar body nuclei, and then you have 2 other nuclei from either the paternal or the maternal side. But this methylation staining indicates that only the mother side is methylated, right? Only the mother side nucleus is methylated, as well as the PP2, the polar body 2 DNA. They are methylated, but not the paternal side. That's because the paternal side has already the DNA mark, the methylation has already been erased at this stage. It gets erased much quicker. That's why, you know, you can use this methylation staining to tell you what is what. Okay, you can tell apart which nuclei is from the paternal side, which is from the maternal side. But since you're taking both, it really doesn't matter that much. But you know, to be precise, you still want to get those identified. That's what they're trying to do. And then let's study the outcome. They did 4 different manipulations in parallel. They're trying to compare which is the best way to correct that disease, that mitochondrial disease. Now, and here in A, it is just the development. You count the embryos at different developmental stages. Although you start with the same number, you want to count. You want to count. You know how many successfully goes through the developmental stages, such as shown in C. Right? You have a reconstructed embryo. That's the embryo after manipulation. What you did the transfer. So that's the look of the embryo. And then you have F1 infants. F1 infant is the direct outcome of this embryo. And those infants will become adult, right? And then eventually those adults will mate with additional other animals to give rise to the next generation. So that's the developmental stages. But that's not the detailed developmental stage. It's just the different generations. They show their experiment design. They show to you that every manipulation can give rise to F1 infant, F1 adults, and the next generation. That means it's, roughly speaking, it didn't have much, major problems there. The reproduction can still occur with this after this manipulation. And in A, what they are trying to tell you is the detailed developmental stage. Okay, this is 2-cell stage indicated in blue, 4-cell stage indicated by green, and brown indicate blastocyst stage, and then those even darker purple or brown indicate the offspring, live birth. As you can see, all the manipulations, no matter it is PB1 transfer or spindle transfer or PB2 transfer or pronuclei transfer, the 4 different ones shows very similar results. And although, strictly speaking, maybe when you compare those manipulations to IVF, the pronuclei transfer is the closest to the efficiency of IVF. But still, the rest of the manipulations are very good, very good. The drop of efficiency is quite minimal. And in B, it shows you the offspring, the developmental stage at blastocyst. As you can see, that when you compare the 5 different manipulations, because you have 4 manipulations in addition to the reference, that's the natural IVF. You can see at blastocyst stage, this PB2 transfer actually generated less efficiency. So when you transfer PB1 or spindle or pronuclei, you can reach similar efficiency of blastocyst stage. But PB2 transfer somehow resulted in a drop of efficiency. So that's maybe you're trying to correct this disease, but you have to consider a lot of practical restriction. If PB2 transfer is not as efficient, maybe you consider that not as an ideal, solution for that mitochondrial problem. But here, I would say 55% is still very good, right? So although there's a drop, maybe it's still acceptable. Now let's study, does it correct the problem? So here in A and B, it shows you something called heteroplasmy. The heteroplasmy, that means you know, when you have a recipient that's a healthy oocyte or zygote that have healthy mitochondria, and now you have a carryover. Carryover, that's the unhealthy mitochondria that you unintentionally put into the healthy oocyte or zygote. So when you have a carryover, you're going to expect heteroplasmy. That means for the same mitochondria gene, you're going to have two different variants. One is the healthy variant, the other is the unhealthy variant. For example, like here, after DNA sequencing, you see like in one of the party is a C, while in the other party is a T. While the rest of the gene are exactly the same, but the mutation is, you have both alleles in presence. Okay, so from that you can measure how successful or how minimum that carryover is. So you can measure heteroplasmy. So here is the result. We're comparing 4 different transfer approaches. As you can see, the PP1 transfer in the F1 generation, that's the direct generation, you have a very minimum carryover. So I'll skip the details of the other two, but you can see the pronuclei transfer, that's PPT here, have the highest heteroplasmy. Okay, so the same thing happens in PP2. So basically, you have the F1 animals to mate with additional animals to generate in the next generation to test, whether this approach fix this problem, not only for this individual, but also for the offspring of that individual. Do you have that advantage? Do you have that, outcome that you're looking forward to? But it's true. Even in F2, you see that polar body 1 transfer give you minimum carryover and minimum heteroplasmy. And this is tested again, not only in the whole animal, but also in different organs, like in brain, heart, lung, liver, and kidney. What in several replicates, what they have shown is still PP1 gave you the highest, the most advantage, least carryover. But I believe in UK today, the transfer, the government approved the transfer is still, the transfer of either the spindle or the pronuclei. Why? I guess it's mainly because of, I won't say it's ethical. In human studies, when you transfer the PB1, it's a polar body. It's not exactly the nucleus of the oocyte. So there will be some additional risk that the animals you don't observe, but there is still some risk there, because after all, that polar body is not oocyte, right? So you take the oocyte nucleus, you put into the counterpart of a healthy oocyte, then you expect least risk, although it may not be the best in terms of minimal carryover, but maybe other risk will be much smaller. Okay, so in humans, they are still transferring the nucleus, the spindle. They're transferring the pronuclei. But here in this animal experiment, it's demonstrated that PP1, if you transfer PP1, that gives you the best outcome. It gives you the outcome you expect it to be. So I hope in the future they can remove their unnecessary concerns and start to test that PP1 transfer, because it gives you the highest advantage. So now I want to give you a summary of somatic cell nucleotransfer. As you know, this was introduced by Sir Gurdon in 1960. That's many years ago. I'm surprised I was surprised when I saw him, because it's imagine how long time it was, how long time ago it was. Now it's 2020, It's 60 years since his discovery. So he was a graduate student, so he must be like 25 or older, right, as a graduate student. And then I see him giving talks, writing papers. His papers often is single author paper. It's just amazing, the human being can be that productive and also have that much energy to pursue science until today, with not winning at all after his his early major discovery. So this is the old technology, but people are concerned about reproductive cloning, and in this country, there's no way that human SENT is supported in any research. So if you want to do that, it better be funded from private company, not by the government. And even if it is from the private company, whether it is legal, it's still a big question mark there. But in agriculture, it's being explored everywhere to expand the premium qualities. Remember, bring back the mammoth. And so a lot of time passed since they were able to use this technology to establish human embryonic stem cell, embryonic stem cell line. That was only 5 or 6 years ago. People say that if you reprogram the cell using somatic cell nuclear transfer, the fidelity is higher than iPSC, than when you use several transcription factors. So you reprogram a somatic cell nucleus by this nuclear transfer, or you can reprogram that nucleus by putting in several transcription factors. They say that this NT-ESC has higher fidelity toward the gold standard ESC than iPSC. And in today's lecture, you learned that this has a unique position in correcting mitochondrial disease. Basically, you just take the genetic material of the unhealthy mom and then put it into a healthy donor. And then that solved the mitochondrial problem. The problem of mitochondrial mutations. Okay, that's all of today's lecture. Group 3, I hope someone of you are present in today's lecture. Identify yourself. Yes.
We're group 2 that presenting on Friday.
Yes, but is there anybody from group 3?
Yes.
Yeah, because I posted the study material for next Friday. I didn't announce in today's lecture, so I want you guys to know that, so that you can, if you want to, you can prepare ahead of time.
All right, sounds good.
Awesome.
Okay. That's it for today's lecture.
Yeah, it's posted on Blackboard or on Teams?
No, it's always in Teams class materials.
Okay, sounds good. All
So
right.
you're free to go if you don't have questions.
Thank you.
Mm-hmm.
Dr. Liu, so
yes.
I was just wondering then, the